just going through our kids, we have Gideon who's 13.
Dr. Scott,
who's 5.
hold it closer. It may sound loud, but
Gideon who's 5.
Now we can hear in the back.
Gideon who's 5. Tree who's 21. We have April, who's my cute wife. We have Colton, who's 2. We have me. And then Tara, who's 19. Tiana, who's 15. And Tori, who's 17. So we have quite a spread. So we have from 21 to 2. And we are a little bit on the crazy side. We had a friend of ours who recently came to our house, and they commented, they said, we just love being in your home. I said, well, that's really, really nice. And I said, why do you say that? She said, it's so calm. I'm like, really? I'm like, where do you live? Because our house is usually pretty chaotic. But when I think about myself, the very first thing I think about is the fact that I'm the dad. And so all of you are here because you're parents, and at least I get that. I understand at least at some level the things that you're thinking about, because I think about the same things with my kids. In addition to being the dad, I have to feed all these kids, and so I'm also a clinical geneticist, and I'm also a researcher. And as was explained, I have an MD and a PhD, but basically the whole idea of what I do is I see kids that have different genetic disorders, and then I try to figure out why they have that, and what causes that. And pretty much if I'm in the hospital, I'm doing that, and if I'm in the lab, I'm also doing the same thing. And what we find is that things that we see in the clinic help us to figure out what we need to do as far as our research lab goes. And so one could say, well, why are you here? So why am I at the 1p36 conference? And the answer is I see a lot of kids that do have 1p36 deletions. And we do research on 1p36. And so it makes sense that I would volunteer to come. But the biggest thing I want to do, if you say, well, what's your goal? Is I just want to answer your questions. So most of what I talk about today would be the questions that I often get from families that come visit me in the clinic. I want to talk about research and stuff too. Hopefully there'll be a little bit of something for everyone. But if you have specific questions that you want to ask, that's kind of what I'm here for. And I'm going to be here all day long, so there'll be plenty of time to ask questions. So one of the very first questions I want to talk about is, why does my son or daughter have medical problems? Basically, children that have 1p36 deletions have an imbalance in their genetic material. And we say, well, what do you mean by an imbalance? These are all the chromosomes that we have in our body. And the ones that are highlighted that's chromosome 1, which is actually the biggest chromosome. And God intended for our bodies to have basically 2 copies of every instruction. And when we are missing a copy, it throws off the balance of how things should be. As it turns out, if you have 3 copies of something, it also throws off the balance. And so we can have problems with kids that have too little or too much of something, and it just shifts things a little bit. If you were to miss the entire chromosome 1, most of the children that would have that would actually not survive to birth. So those would be ones that would usually die in utero and usually probably pretty early on in pregnancy. But again, we're here, and so we're not talking about the entire chromosome 1, we're talking about just that tip piece, the very top. And because it's only that tip, that allows, again, the function or the development of the body to be sufficient so that kids can get all the way through pregnancy and then be born and then live beyond that. But it's, again, only because it doesn't take so much of an imbalance. It only affects that tip part. Often I tell my families when they say, well, I don't understand this balance part. So often in the clinic I say, well, it's just like making cookies. It's very, very similar actually. And so I asked my wife, I said, when you make your awesome chocolate chip cookies, what do you use? She said, well, I can tell you the ingredients, and if you want to know how I do this, I said, just give me the ingredients. She says, well, for our particular cookies, we use flour, white sugar, we use brown sugar, eggs, butter, vanilla, baking soda, and chocolate chips. And I said, perfect. And you can imagine that if you were to shift the balance of any one of those things, you would have a little bit different chocolate chip cookie. There would still be cookies, and you would recognize them as cookies, but there would be a different flavor to them, or there'd be something that would be different than the way my wife usually makes them. And basically, that's the same kind of thing we're talking about. It's not that we're removing like an entire ingredient, like chocolate chips are just gone, or the flour is just gone, but we're shifting the balance of things. So we're not putting quite as much sugar in, or maybe, if there's 2 eggs and there should be 3. That's the kind of small shifts that we're talking about. When you say, well, what are our ingredients? So how to make a person? We make a lot of water. We have proteins, and we have fat, we have minerals like our bones, we have carbohydrates that actually come in lots of different parts of our body, and then we have DNA as well. When we say, well, what causes a child with 1p36 deletion to have difficulties, we're mostly talking about the proteins. At least that's what we concentrate on mostly. And he said, well, how many proteins are there in the body? No one exactly knows for sure, but there's probably about 100,000 different kinds of proteins that make up our body. And those proteins are coded for about 20,000 different genes. So that's that DNA part. But we're talking about 645 genes. So out of 20,000, it's a portion. It's a significant portion, but it's certainly not the whole. It's not a whole chromosome 1, which would be literally thousands of genes. And so, why do some children with 1p36 deletion have more medical problems than others? This all comes down to the fact that each 1p36 deletion can be of a different size and also may affect different genes. So this is the top part of chromosome 1p36, just like a picture of it. And often we talk about kids having terminal deletions. And a terminal deletion basically means it starts at the top or the tip of the chromosome. And those terminal deletions can, again, come in many different sizes. So, for example, if you just take out that very, very, very top band, the very tip, that tip would contain about 99 different genes. But you could go a lot farther and take up a bigger portion, and that might include 186 genes. And so you can see that certainly it would be a difference if you're just affecting more genes just the farther you go in on a terminal deletion. There's other kinds of deletions besides terminal. The other one is called an interstitial deletion. These are ones that we're seeing more and more commonly because of the techniques that we use allow us to see them, where if you said 10 or 20 years ago, we just didn't have the ability to look for these kind of changes. And so an interstitial deletion, instead of hitting the top, it actually takes out a piece from the middle. And those interstitial deletions also can be of different sizes. So in this particular case, that would create a problem for 72 different genes. But again, they'd be completely different genes than the ones that were at the tip. And so you begin to see that everybody has kind of a different size and may actually affect more or less genes. And that's one of the reasons why different kids look differently, even though they have the same syndrome. The other thing they have to remember is that kids just don't look alike. They're just different. A lot of you have more than one child. I have 7. All of them are all different. At one point, I remember when our second daughter was born, she looked exactly like the first one. And I remember just for a split second, I'm like, wow, this is going to be really boring if like all the kids look and act exactly the same. Now, as it turns out, she ended up having red hair, and she kind of acts like she has red hair. And, she's a completely different kid. You all see that, and we recognize that. And yet, sometimes, people like to try to group everybody and think, well, there should be the same because they have the same disease. And certainly that's not the case. So there's just a lot of genetic background that also makes a difference between one child and another. And we should expect that just as much in 1p36 deletions as we would in anything else. So if we mix those two, a different deletion and a different genetic background, we can certainly say that every child with a 1p36 deletion is unique. They're not just unique in who they are, but they're actually genetically unique too. And so every one basically can teach us things, or we can learn, but that's also the difficulty that we have when families come and visit us. Because one of the big questions families have is, well, what will this mean for my child? We try to answer that, but most times, especially when they're very small, I say, I can only give you a feel for what it might be like, and then we'll just have to see how things go. Question number 3. How do doctors know which region of the 1p36 gene is deleted? This is a really complex question because it depends on when your 1p36 deletion was identified. And so some of you might be able to come up and say, well, my child is now 15 years old, and he was diagnosed when he was 1. Chances are you used certain techniques in order to be able to find that. If you said, well, my child was diagnosed 7 years ago, it would actually use another technique that was available then that wasn't available 15 years ago. But if you said, how are 1p36 children identified now, almost all of them will be identified using one technique that's called a microarray. So I'm going to talk about microarray analysis just really quick because this is how we do it now. And this is why we can do such a detailed job of identifying exactly what the deletion looks like. I was just put in right now to say, if your child was diagnosed 20 years ago, could you use this technique to identify exactly what piece is deleted? The answer is yes. And so often we see kids that will come back, and because of wanting to know more, we'll sometimes suggest, well, why don't we get a microarray analysis and see exactly what we're dealing with. But let me show you how that works. So the very first thing that we do is we take a blood sample. And almost everything we do genetically seems like we do on either blood or urine, but mostly it's blood. And if you say, well, those blood cells that we're giving to the doctor or to the laboratory, the only ones that we really use are the white blood cells. Most of the red blood cells don't have a nucleus, so they don't have DNA. Instead, they're really great for transporting oxygen. But we're interested in the white blood cells that have a nucleus, so we can get the DNA out of them. So of all the sample, we take just a fraction of those that we get the DNA out of. This is one of the reasons why if a child has a transfusion, it's no issue for us because we only transfuse red cells, and so we can do a genetic test basically any time a child has white blood cells that we can get out of their blood. So basically, whenever you get a blood transfusion, it's either screened out or the white blood cells are killed, and this doesn't become an issue for us. So anyway, once we have the DNA, then what happens next? This is where it gets a little bit high technology, and I won't explain everything, but let's just say it takes a laser scanner to do this, and those laser scanners usually cost somewhere around $250,000. And so some people have them and some people don't. Not every laboratory is going to be able to do this test. Once you buy a laser scanner, well, it's good forever, theoretically, but it's a big investment. That's why you don't have these type of labs done at your local hospital. Instead, they'll have to send it to a big hospital or a big laboratory that has a laser scanner. The second piece of the technology that we use is this little tiny chip. I don't know if you can see it, but basically it's a piece of glass, and if you can really look really carefully, there's little tiny rows on it. That little piece of glass actually will have thousands of DNA probes. Each one of those probes is especially designed to test one piece of the DNA and say, is it there, and how many copies of it are there? This is a technology that's pretty cool. They actually use a printer to print these chips. So there's a special printer. It looks probably like 1/8 the size of this room. And they literally use an inkjet printer, and they print the DNA on this chip, just like you do at home, and they actually make these chips. The chips will cost sometimes hundreds, sometimes even thousands of dollars. And so the test is not cheap, but it's amazing what we can do. So this is actually the raw data from a chip that has 1 million probes. So it's going to check 1 million different pieces of that DNA and tell us whether they're 1 copy, 2 copies, or 3 copies. If you blow it up, like under a microscope, you can actually see that every single one of those things is dots. So they're all dots. And this has been scanned by a laser scanner, and so the dots have different colors. And so we have some dots that are green, some dots that are yellow, and some dots that are red. But remember, every one of those dots represents one specific part of the DNA. So if we just take a look at those 3 spots, what that means, or those colors actually tell us how many copies there are of DNA in that area. So a green means there's one copy. Yellow would be normal, having two copies, one from mom, one from dad. And then red would mean you have three copies, which means somewhere along the line there's been a change and you actually have extra DNA material. Now, you can imagine it's not very easy. I can't scan this with my eye. I have to have actually special software that can deal with a million different pieces of information about my one patient. So what we do is we put all this into a computer that basically allows us to see it as we see it on a chromosome. Now, this particular patient doesn't have a 1p36 deletion. Instead, they have a deletion in another part of chromosome 1. But it's pretty easy to see. I mean, all the black dots mean there's two copies. All the green dots means there's only one copy. And so you can really easily see where that deletion is. So if we just kind of blow this up, you say, well, I want to know where it starts. I want to know where it stops. And then I want to know what kind of genes there are inside. This particular array comes from a child that I saw in clinic who had some difficulty with autism. And there was also some concerns that there might be some other behavioral abnormalities. When we found this deletion, we recognized that there was a gene in it called TSNAX. That gene has been linked to autism, and it's also been linked to schizophrenia. And so when we saw this, we said, well, this is probably the cause of the behavioral problems and such that we're seeing in this child. The same kind of principle applies, or could apply, for kids that have a 1p36 deletion. And that's kind of what we're going to be talking about today. But anyway, super powerful. We can say exactly, like down to almost the very letter, where a deletion begins and where it ends. What does this look like to doctor? So a doctor doesn't actually use that computer thing. Instead, that's mostly done by a laboratory. But the doctor will receive a printout or a report that looks like this. And some of you may have these. So this happens to be from Baylor College of Medicine, which is where I work. So this is our laboratory that does this particular one. And it'll give data on the person and their birth date and all that. But the most important thing for us as a doctor is we want to know what is the exact region that's deleted on chromosome 1p36. And we have special ways of exactly noting the portion of the chromosome. But the biggest thing for me is I want to know these numbers. Because with those numbers, I can go down and say exactly what DNA is missing. And so for me, with this little bar of information, I can tell you everything about this child's deletion. I can tell you how many genes it has. I can tell you what genes they are that are deleted. This is a gold mine. And a lot of you probably have one of these, or your geneticist or your doctor has one of these, and it's super helpful. And then down here, there'll usually be an interpretation where they'll say, well, this means this, because these reports go out to all kinds of different doctors, and so sometimes they don't know that a 1p36 deletion means a certain thing, so we'll tell them about that. So that's the most important part for me. Is each medical problem caused by a deletion of a specific gene? In other words, can you say, well, my child has a heart defect, and that's because in his deletion he has this particular gene that's missing? That's a really, really good question, but it's not a yes or no question. Instead, I'll have to kind of explain why I can't say that right off the bat, yes or no. Let's go back to that terminal deletion kind of idea. Okay, about I'd say maybe 8 years ago, we began to have the technology like arrays to be able to see different portions of chromosome 1p36 that were missing. Shortly after we had that technology, it became clear that we had terminal deletions and we had interstitial deletions. But if you think about terminal deletions, they definitely can get bigger, so some of them are small, some of them are large. But if you say, well, what do terminal deletions cause? They cause a whole number of different medical problems. And you can probably go through this list and say yes or no for your individual child. But what we saw as geneticists was that, well, one of the first things people wanted to find out was what's the smallest deletion? What's the smallest deletion that can cause basically all these problems? So they did research, and they found that there was a very, very small piece on the top of chromosome 1 that could basically do all these things. That is about 6 million base pairs long. So there's 6 million letters in that region. And if you delete that, it would be sufficient to cause this, right? Basically all the problems. Now, that's like a great answer, because now we knew where the genes were. But then there was another group who said, well, actually, if you have a middle deletion, it can cause almost the exact same thing. In fact, almost everything can be caused by an area that doesn't even overlap that tip. And so all of a sudden we were saying, well, we're looking at maybe 2 different diseases here. And so they said, well, these kids don't even have the same look. Maybe this is a different syndrome. But you have to remember that there are some kids that are deleted for both of these critical regions. And so some of them have a big deletion. But anyway, we knew from this paper that there had to be at least 2 genes on chromosome 1p36 that caused almost all of these different problems. And so, depending on what your deletion is, we may be looking at 1 gene that causes it, or we may be looking at 2, or 3, or 4. It just depends. So what we started doing in our lab is we said, well, we want to know not just if there's 2 regions, we want to know how many regions, how many genes cause things. One of the easiest ones to do was to say, well, what about cardiovascular malformations and cardiomyopathy? Just out of show of hands, how many of you have children who have one of those 2 problems? Okay, we expect about that. So the estimates are about 71% of the bigger deletions will have a cardiac defect, and about 28-ish% will have a cardiomyopathy, which could be left ventricular non-compaction, if you've heard that. Probably some of you said yes. Yeah, that's kind of a new thing. Or it could be dilated cardiomyopathy. Anyway, but we wanted to find out, well, how many genes are there that do this? How many genes are involved? So I have a graduate student. She actually looks much nicer than her picture. Her name is Hatisha. And I said, Hatisha, I think we could do this. Would you be willing to help me to analyze all the data from as many kids as we can find with 1p36 deletions and see if we can map all the different genes that cause these 2 problems? She said, sure. Which was a good thing, because it was a lot of work and I didn't want to do it all. But anyway, we said, well, let's lay some ground rules first. So we're going to look at only isolated cases. So some of you may have children that have a piece of 1p36 that's missing, but it has a triplication on another chromosome. There's a part that's, or maybe have a balance that they're deleted for one part, but they're triplicated for another part. Those become very confusing for us, because we're like, well, we can't tell which piece is doing what. So we said, well, let's make it easier for ourselves. We're just going to do isolated. They have to have microarray data, because we want to be able to know exactly where their deletion is. They've got to have, well, one of those 2 problems. If we did that, where would we find those patients? So some patients come to us. So we have patients that call us up and say, we want to be part of your research study. We have people that are patients that are referred to Baylor, to our medical laboratory, for microarray analysis. So we could look at them. We also have databases as geneticists. One of them is called Decipher. It's a database where all doctors all over the world can put data from their patients, including patients that have 1p36 deletions. And we said, well, we're going to search the literature and just try to see how many we can find that have already been published, because sometimes it's just a matter of putting all the stuff we already know together. So when we did this, we actually found that there were surprisingly few cases. Part of the reason for that is because the arrays are relatively new. And part of it is just because we don't have enough parents that are on board yet with the studies that we do. And so we said, well, even with the few cases we can find, can we find and make some progress? So this is all the cases that we could find have cardiovascular malformations. And basically the different colors are the different databases or literature that we used. But you can see that we have patients that have terminal deletions and interstitial deletions. They can be big, they can be small, they can be special where there's like a gap and they're missing 2 pieces, one over here and one over here. But if we said, well, how many regions could be responsible?
Well, we can outline five different patients where everybody else lines up over top of one of these deletions. So you can say there's 5 critical regions and they don't overlap. That means, like, right off the bat, there's got to be 5 genes that cause cardiovascular defects, at least. Or at least 5 genetic regions that cause this. So then I said to another graduate student, I said, hey, you know, Atisha's been working really hard, and we've found all this stuff. So Tyler, I want you to now go in, and I want you to find out what are the genes in each one of these regions, and are there any that are known to be hooked or linked to cardiovascular problems? So I need you to do that for me. And he said, not a problem. There's all these great online tools that will help us do this. He says there's one called Gene Distiller, which sounds like kind of fun, you know. Anyway, basically it's the idea of you take a bunch of genes and you stick it in there and it pops out the ones that are the most important. Like, that sounds great. So how does Gene Distiller work? Basically what we do is we take the region that we're interested in. In this particular case, it's from the telomere, which is the start. So that's the very tip. So it doesn't even have a number. And I'm going to go a certain number of letters into that, into chromosome 1. You can begin to see why it's important that we have exact numbers, right? But this will tell us how many genes there are in this area, and then also helps us to do even more than that. We then said, okay, well, what data do we want to have it pull out for us? So what we do is we go through and we pick the data that we want. But it has some important things in it that are worth pointing out. So for example, there's a thing that we use as geneticists called OMIM, Online Mendelian Inheritance in Man. It's basically, again, a big database for geneticists and doctors where we put patient information in when a gene is known. Then there's also, on this other side, this is the mouse version of that. So this is kind of the human version where doctors put in stuff, and this is where mouse researchers put that same data. And so we're going to look for any human and any mouse gene, because mice are very similar to us, any gene in that region that might be related to heart. So when you do this, you get a ton of data. This is just the start of one data for one gene. Okay, this gene is called SKI, but this is the human side of the data. And then all this is the mouse stuff. Just kind of goes on and on. And so what we did was we said, well, yeah, it's too much to read. Let's put it all in a spreadsheet. So we put it all in a spreadsheet. And then we said, well, let's just use the spreadsheet, and we're going to look up a couple different words. We're going to search for cardio, cardiac, heart, and just say anywhere in the spreadsheet do we hit those terms. And when you do that, you end up with some genes that come out positive and some genes that don't. Then we have to actually go by hand and read all the stuff about those genes. And then we say, well, are there any that we believe really might be the cause or might be causing this problem? So when we looked at the 5 different regions and we did all that work, we then said, what are the genes? So we found some genes for the first region, this DVL1 and SKI, for example. There's another gene for the next one. There's 2 genes for the next one, 3 genes for the next one, and another gene for the last. Now, all of a sudden you begin to see how the complexity of this, because we're saying that not only might a person have a big deletion that affects all of these genes, but even ones that have really small deletions, they may have one gene that's affected, or they may have 3 different genes that all might be contributing to that heart defect. And so all of a sudden it becomes more interesting, but at the same time it becomes quite complex. We're not talking about one gene. I'll just tell you that I have geneticists still, people who should know better, that will come up to me and say, have you figured out what the gene for 1p36 is yet? And I just smile and I say, not yet. Not yet. Because it's just, we're not talking the same game. I mean, it's totally different. We think about this as a disease where every child is going to be different. And so we're not going to find one gene, we're going to find many genes. And then it's a question of what is each one of those genes doing. But this is a hard problem. Okay, what if you did cardiomyopathy? You actually don't have a lot of cardiomyopathy kids that have that kind of data. Again, we have many more kids, but we're being very selective in what we make these regions based on. I mean, they have to be specific things that we can use, and so we're being very, very snicky as far as our selection. But if you said, what about cardiomyopathy? Well, there's still two non-overlapping regions for cardiomyopathy too. So there's got to be at least 2 genes out there. We do the whole same process over again for those 2, and we find 3 different genes that might be important in that first region, and we find 2 more genes that might be important in the second region. So even here, we're talking maybe up to 5 different genes, and we're just beginning, because there's still many genes that we don't even know what they do. These are only the ones where we actually find something in a human or something in a mouse that tips us off. It's getting kind of complex. So, it is a really good question. Some medical problems may be caused by loss of a single gene. True, could be true. Other medical problems may be caused by loss of several genes. Probably even more true for most of our kids. And also, the same medical problem can be caused by different genes in different people. Now, that becomes harder because if you're going to treat it, you almost need to know, well, what is causing that in this child? And then you have to do something about that problem. Okay. Does knowing the genes help people? People always ask me this too. Like, oh, that's really great that you're doing this. Does it help anybody? The answer is it helps us a bunch. So this kind of data is super helpful for a geneticist. Why is that? Well, let me just give you a for instance. So basically I'm saying it helps make doctors make better medical decisions. Let's say you have a child, brand new child, just born, and we know even prenatally maybe that this child has a 1p36 deletion. One of the things that we must decide is, does this child need to be screened for cardiomyopathy? Now, I don't know about you, but this is a big question for us. It's how often do we need to screen and do an echocardiogram in order to be able to catch the cardiomyopathy? Is it going to be something that's going to present early? Is it going to present late? Does it make a difference on what the deletion looks like? But anyway, most of us, as geneticists, we kind of say, well, we'll just have to screen them. And once every 3 years, once every 5 years, we'll screen the child. It's probably not the best way of doing things. But could we at least know who we should screen? Like, does everybody have to get screened? And so that's when we start to say, well, if these truly are the only genes, if they are the only genes that cause cardiomyopathy, then you have a child that has a deletion here. Should I screen them for cardiomyopathy? The answer is definitely yes, because they have all those genes affected. If a child has a deletion on the other side, do they need to be screened? Well, at least for now, we know of no genes, and I can't find a single case of anyone who has a cardiomyopathy that's involved with that region. So I would say we could skip the echocardiogram that costs, I don't know how much these days, but it's a lot. And we don't have to have the parents come in either for those constant screenings. Not only that, I can sit down with them and say, you know what, you've read about cardiomyopathy in 1p36, and you're not going to have to play that game. That to me is super powerful and useful. And so beginning to understand the genes at least helps me do a better job with my job. So I'd say no. Identifying the genes also is the first step to being able to develop new treatments. So if we're going to be able to figure out what we're going to do about this, we need to know what the genes are. The second step is actually probably even a little harder, which is to learn what those genes do in the body and then how do they do it. That's the part that takes actually the longest time of anything. So the next question is, well, how do scientists actually go about figuring out what a gene does? So let's say I have a gene on chromosome 1p36 and I just want to know what it does. Is it involved? One of the ways we do is we actually sometimes learn from individuals that have these deletions, or sometimes individuals that have mutations within specific genes. So there's a great example of how this just happened just literally months ago for a gene that has to do with cardiomyopathy on 1p36. So remember, these are the genes that we mapped out. Well, one of these genes called PRM16, recently, like 2 months ago, there's a group that found that there were changes or mutations in this gene in individuals who do not have 1p36 deletion syndrome, but do have cardiomyopathy, just all by itself. And specifically, they have left ventricular non-compaction, and this may be the most important gene for that particular problem because it's sufficient to cause it. In other words, if you have this, it could push a child into having cardiomyopathy. Does every child that has this deletion have that? The answer is no. So sometimes we see that this gene can also be affected in people who are perfectly normal and never have any heart problems. But it would definitely be a risk factor for a child to have that particular gene that's deleted. This is really the first cardiomyopathy gene that's ever been, if you would, proven for 1p36, and it's happened 2 months ago. How many of you knew about this? Okay, that's what I thought. I didn't know about it either until about a month ago, because it takes time.
So does that mean it would be useful for our doctors? Is this published yet, or like to know if we should be having our children screened?
So the question comes, is this been published yet? Yes, it has. So it came out 2 months ago, and a friend of mine actually sent it to me about a month ago and said, have you seen this? I was like, no, I was a little busy. But it becomes super important. If you had a brand new baby who was born and they had a deletion of this gene, your doctor should put the 2 and 2 together and should screen that child for cardiomyopathy. And so I would say yes, this is an actionable thing. So we would do this. And right now, with that information out there, as a geneticist, I want to say I'm liable because that sounds really like lawyer. But basically, I would feel really, really bad if I was to miss this. This would be my fault because now I should know this and I should take action accordingly. Okay? Don't forget, though, that just because you're missing this gene doesn't guarantee you will have a cardiomyopathy. We just know that it is one of the major players. And therefore, we should be screening and thinking about this. Because remember that brother thing where different brothers look differently? That genetic background makes a difference. Okay? Because some people have errors with this gene and do fine. Others have errors in this gene and get left ventricular non-compaction. And there's a question whether they'd also maybe be in certain individuals with cardiomyopathy. Yes? Is there another name for this gene? Almost every gene has more than one name. This is the official name right now. But this is a relatively new gene. There's not a whole lot published on it. So every researcher I've ever talked to that works on this particular family of genes refers to it this way. So I don't think you're unlikely to have caught it in a different name. Yes?
If you're having screenings every 3 to 5 years, because there is a current cardiomyopathy, is that indefinitely, or is it some point when you think you would develop or not develop?
So the question is if we're screening every 3 to 5 years for this, is there a time when you stop worrying about this? That's a good question, and one that I do not know. I've asked many people, I said, what is the right thing to do? And the answer is, no one seems to be able to give me the right answer. What will it take? It will take us to have enough kids with 1p36 deletions that involve that gene, and hopefully not a whole bunch of other ones. And then we'll have to sit down and say, how many of them get cardiomyopathy, and when do they get it? Now that's a lot of data. When I can do that, then I might be able to say, these kids will always present within the first 3 years. So after 3 years, you don't have to do this. The problem is we don't have that data. But it's something that we really wish we had, because it would make a difference, again, on how we treat patients. The bad part is that right now, what we have is we have data on a whole bunch of kids with 1p36 deletions, but we don't have them mapped to the kind of detail that we need to be able to say, this is what you do when this is deleted, and this is what you do when this bigger piece is deleted. It's going to take a lot more. Now, if you said, well, could we do that? Like, could we make a database? Could we make a database as a bunch of families and do this? The answer is that has been done with other deletions, and it would be very valuable. We are interested in doing that, and that's why we collect families. In other words, we get the the data from like the deletions and your family history and clinical history. We try to put it all together. But yes, could this be done? Yes. Is it done for other genes? Yes. And it's very valuable. Some of you probably have heard of Unique. Probably yes, I imagine a lot of you. Unique does a great job. I mean, I've been very impressed. It's a database that we use for all these disorders, and they collect families or family information, and then they try to distill it and then disseminate that information. They've done a really great job. I use their stuff all the time in genetics clinic. Why? Because they've caught the vision of what you can do with a database. So anyway, the long story is, that's a really good question, and I don't know the answer. But it could be done. It could be done. But we would want to do it on that kind of level, where we do it for that specific gene. Because I'm afraid that what will happen is, is we could say this will always occur within the first year, and then when a child has a bigger deletion that also includes these genes, it could be a different game. And so we have to do it and do it well. Okay, I'm going to go on, but I'll take more questions at the end. Does that sound good? Okay. All right. So sometimes we can learn things from individuals that have mutations within a single gene. Sometimes we also can learn from animal models. This is the time when I get to tell a story. So I want to tell you the story of how I began to do 1p36 research. Okay, I've been a doctor for a really, really long time, and I saw kids that have 1p36 deletions, and so it's in the back of my mind. But it's one thing to see kids in a hospital, and it's another thing to say, I'm going to dedicate my life to doing this. Okay. Most of us who dedicate our lives to doing something is because we either have a family member or we were in a lab that was interested in this. I had no connections to 1p36 at all until I got this mouse. So it all starts with a mouse. This is a normal mouse. It has normal eyes. This mouse does not have normal eyes. This mouse has microphthalmia, super small eyes. Once upon a time, there was a researcher at Baylor who joined a group where they were just mutagenizing all kinds of genes to see what they did. And they did this in mice. They generated a whole bunch of mice that had abnormal eyes. And they said, we're going to study those abnormal eyes. And they did so for a little while. But then they could not get funding to do this any longer. And one of the researchers decided to move from Baylor, which is where I work, and go to a different university to do more clinical work. Well, as he was leaving, he came to me and said, Daryl, you're a relatively young and naive person. Like, yeah. And he said, would you be interested in some of my blind mice? And I said, I don't know. How'd you get them? He explained how they were generated, and he said, I don't know the genes for them yet. I don't know the genes that cause this, but we're getting closer. Would you be interested? So I talked to my advisor, and I said, I've got these blind mice things that someone's offering me. Should I take them? And he said, well, sure. I mean, eye research is great. I've never done anything with eye research, ever. And he's like, oh, you should take them. So I said, sure, I'll take your mice. So this is one of the mice that I took from him, was this one. Now, we didn't own the gene for it yet, but that was up to me then to find out. So after a lot of work, we found that the gene that was missing or hurt in this mouse was a gene called RERE. Doesn't matter what, I'll talk about what RERE is. But basically, we could only find the change, and we didn't know much more about this gene, but we knew it was the cause of the eye defects. If you say, well, how does this work? I'll just give you a little quickie, okay? So we have this thing called sequencing, where we can actually sequence or determine the letter code of the DNA. So in this particular case, this is wild type, and it has a T, which is represented by a red up kind of thing, and these mice don't have that, they have an A. And on the other gene, or other allele, so this, let's say, comes from dad, this comes from mom, there's another change. This time it was supposed to be a T and it ended up being a C. Those changes make dramatic changes in this protein. So one of them makes it skip. Exons are the part of the protein that, or part of the gene that codes for protein. So this one makes it hard for the body to put this piece of protein together with this piece of protein. And this change makes this piece of protein not work. And so we basically had 2 changes in these mice, all in the same gene. Now you say, well, that's very, very nice. But why does that mean anything to 1p36? Well, this gene, bigger version, this gene is actually on chromosome 1p36. We label our mice according to the mutations that they have, but this gene hits right in here. When I found that out, I said, now this is a really interesting gene. I know this kind of gene. This kind of gene does lots of stuff. It doesn't just play an eye. This is the kind of gene that can play in a lot of things. In other words, it may be involved in lots of developmental problems. So I said, well, I know that kids that have 1p36 have eye and vision problems, but they don't have microphthalmia like these mice do. So this is a little bit more severe. But we're also pushing this gene a little bit. These mice are pretty bad for this gene. I said, what if they had something else? So I said, well, this gene also happens to be right in the area that causes cardiovascular defects. What would be the chance? Could these mice also have cardiovascular defects? So I sent someone in the lab and said, I want you to find that out for me. I want you to look at mouse hearts and tell me if they have heart defects. So probably a lot of you haven't looked at a mouse heart recently, but let me just kind of give you a guided tour, okay? So this is a mouse heart of actually not a newborn mouse, but actually a mouse embryo, because it's easier, they're smaller, and we can look at their hearts a lot easier. And so this is an embryo heart, but you can tell this is right ventricle and left ventricle, and this is right atrium and left atrium, and it looks a lot like a human heart. I mean, they're all the same parts. So there's also a couple things I can point out specifically. This is a descending aorta. So this is the tube that takes the blood to most of your body. This is the ascending aorta. So basically it takes left ventricle blood, it goes up one side of the aorta and then goes down. So it actually kind of goes like this, like that. So this is the 2 edges or 2 ends of that tube that we're seeing in this mouse. And then this is the ventricular septum, which divides the right ventricle and left ventricle from each other. And it should be solid, no holes. This is a normal mouse, and it looks great. I mean, this is an awesome heart. When we look at our mice, they don't have such awesome hearts. So when we look at their descending aortas, they're on the wrong side. So instead of going up and around like that, they're going up and around like this. And you're not supposed to do that. So that's in the wrong spot. That's called a right-sided aorta. Bad. When we look at where the ascending aorta comes out, it's coming out of the left ventricle, it's coming out of the right ventricle. That's bad. That's putting the blood in the wrong direction. So that's called a double outlet right ventricle, which is bad. Then we look at their septum, and this is easier to see in this mouse, but you can also see that there's a hole between the right and left ventricles in these mice. So this is a ventricular septal defect. Now you say, well, those sound familiar. And the answer is, they should. Some of these defects, especially the ventricular septal defects, are probably seen in a lot of your kids, right? This is one of the most common defects that we see in kids with 1p36 deletions. So all of a sudden, you have my complete attention, okay? Because now we're finding things that are really cool. So I said, well, we know eye and vision problems. We know cardiovascular malformations. So we have the mice. What about other things? So it's also very important, or it sits in a region also for cardiomyopathy. Do these mice have cardiomyopathy? So I said, grad student, go find out if these mice, when they live, because most of them die right around birth. These are not happy heart defects. So most of them die right around birth, but there's a couple that survive. Take the survivors and find out if they have heart problems with their muscle. So this is a happy muscle from a happy wild-type mouse, and it's just red, and it looks like heart should look. These are ones from a mouse that has this gene missing, or at least mutated, and it has all this fibrous, nasty purple stuff. So this also is not right. And then they came back to me and said, wow, Dr. Scott, Like the mouse's hearts are huge. I'm like, very good. Because that's, again, another thing that we think about is having the muscles either be too thick or too dilated. And they said, yes, some of these mice have huge, huge hearts, but not a lot of them live. And this is also probably one of the reasons why. So now we say, okay, well, maybe it plays a whole role in cardiomyopathy. Well, we're on a roll. Why don't we just keep going? So we said, well, do they have postnatal growth deficiency? So in other words, after birth, do they have a hard time growing? And the answer is yes. So these are all their wild type and happy litter mates, and these are the guys that don't grow. In fact, their growth just kind of plateaus. They stay small forever. Do they have brain anomalies? Do they have cognitive impairments and behavior problems? Now, this is harder because our mice are sick, and so we can't do some of this. But we can look at their brains and say, are their brains normal? So we looked at that. And they have small brains. Even before they're born, their brains are too small. And they also have just some really weird abnormalities. This is a part of the brain that helps control movement. And so you're supposed to have 1, 2, 3, 4, 5, all the way up to 10 lobules. These guys are just missing some of them. So they have pieces that are just gone. And this is supposed to be a crack here. No crack really forming there. So their brains are not only small, but they're also abnormal. Now, does that happen in 1p36? Not exactly. But again, we're pushing these mice a little bit farther than some, right? We're making them even worse in this gene. Do they have cleft palates? So how many of you have kids that have clefts? Not so common. Not nearly so common, but yes, definitely there, right? And so we said this is a normal mouse. This is if you were to cut the mouse like this. And so you're looking at the nose. It's kind of the nose. This is the palate, top part of your mouth. And this is the mouth. And our mice, they have a break in that palate. So they're basically their mouth is hooked up to their nose. And so they have cleft palate as well. Like, okay, what about hearing loss? I like hearing loss because I used to do hearing loss research. And I said, find out if these mice are deaf. The way we do that is really cool. We go down to the mouse room, and we have this little box. And this box, you can't hear when you click it. It's ultrasonic, but it sounds like an air horn to a mouse. So you put this thing in the box where the mice are, and you click it, and all the mice just freak out, right? If we did this in this room, all of you would jump off your chairs, right? Anyway, the mice do that too, and well. Normal mice do. But our mice just go about their business because they can't hear well. And this is a more specific test. This is actually the same test that a lot of your kids have had in ABR. And they don't hear nearly so well. So this is good hearing down here. This is bad hearing up here. They hear really bad. Kidney problems. Kidney problems are not so common in 1p36, but they definitely exist. So I said, what about kidney problems? This is a normal mouse, has 2 happy kidneys, and this is the adrenal glands up here. Our mice are missing a kidney, a lot of them, and some of them are even born without kidneys. And it's hard to see, but if you put a little thing around it, you can see them pretty easy. Now this is again above and beyond what we would see in 1p36, but this gene also is involved in kidney development apparently as well. So all of a sudden we find one gene that, at least in mice, hits almost everything that we see in 1p36. And so it caught my complete attention. And so all of a sudden I became a 1p36 researcher. Why? Because we'd always been looking for this. And I knew that lots of people had. And so we think that this is probably a major gene to cause some of these problems, or at least we would hope so. So one could ask, and people ask this, Dr. Scott, do you really believe that RERE causes 1p36 deletion syndrome? And the answer is no. Don't forget, not all kids with 1p36 deletions even touch this gene. Okay? So what can I say? I would say deletions of RERE may be sufficient to cause some problems, maybe. We still don't have enough proof because we'd have to find mutations in RERE. But it may also contribute to a lot of other problems. And it may be that there are other genes that look just like this. And they're all kind of working together, a little bit here, a little bit there, to kind of push kids to having problems. And so we think that this is a an important gene, but we don't know how much it does just yet. Okay. Switching gears. Is there hope for a cure? Or is there hope for new treatments? Often I have parents that ask me this. I would just say a cure would be super difficult. The reason for that is that if you say cure, that means you're going to take everything away and make it all better. The only way to do that would be to actually put back that piece of DNA into every cell in a child's body. And we haven't even come close to being able to do that. Okay? But that's what a cure would look like. And so a cure would be super difficult. I mean, it's not even fathomable to us at this point. Plus, you have to remember that it would have to be done super early. So most of the things that are wrong happen before a person even realizes they're pregnant. I mean, all these things are forming already. So if you say, I figured it out by 1 month of age, we're already there. Heart's already beating. You know, all these things are already forming super early. And so to say, well, my child's now 3, can we change that? The answer is no. I can't change those things. It would have to be changed super, super early. So then the other question is, well, would new treatments be possible? In other words, could you find some things that you could treat? That one I'm a lot more optimistic on. Could you do this? The answer is maybe so. What would we require? Remember we talked about how we first have to identify the genes, then we have to learn what the genes do, and then have to learn how they do it. This is a hard process. It takes a long time to do that. But we begin to do that. We're thinking about this. Mostly we're focusing on the how they do it. So remember that cardiomyopathy, so in our mice? This is something that doesn't happen at birth, but by 3 months of age for a mouse, which is, I don't know, like teenagehood maybe, at 3 months of age, they get this. So this is a postnatal problem after birth. So we say, well, could you correct that in a mouse? And then could that be useful in correcting that in a human? We need to know what this does. This is when it gets kind of confusing and overwhelming, but bear with me for just a second. RERE is part of the vitamin A pathway. So with vitamin A is in all kinds of different foods, right? In our bodies, we take in vitamin A, it gets absorbed in our intestine, and then it gets transferred or converted to this thing called retinol. Retinol then comes into the cell by a transporter called STRA6. It then gets converted to retinoic acid, which then goes into the nucleus of the cell, and then starts to turn on all kinds of different genes. RERE is part of a group of proteins that helps turn on those genes when retinoic acid is there. Now, if you say, well, what about all these other genes? STRA6 causes cardiac defects, causes eye problems. Those kids look almost exactly like our mice. If you have other genes, like this gene or this gene, they also cause cardiac problems. And so they're all kind of working together. But anyway, RERE is here. If you said, well, if RERE is there, and there's a little bit of it that's still there, right? It's not completely gone. Only one copy of it's gone. Could you pump more vitamin A into this pathway and get all these genes to turn on, even if this wasn't working so well? There's some danger in that, because if you have too high of vitamin A levels, it can also make things go wrong. But the answer is yes, maybe we could do this. Maybe. It makes sense that you could try. So for example, one of the things that we'll try with our mice is to give them or their moms more vitamin A and say, can we stop this? Because if we could stop these kind of processes in our mice after they're born, could we do the same thing for a selected portion of individuals that have the same kind of issue? Possibly. Hearing loss. We've done some more tests, and these mice have normal ears. So they look perfectly normal, but there's something that's not functioning well. So when we get adult mice and we knock this out, so we take the gene out when they're adults, they get hearing loss. And so that means that there's something that RERE is doing after birth that makes a difference. So again, you could say, well, if we gave vitamin A or if we gave something, could we get kids to hear if they had this problem? Could we do that? The answer is at least it's structurally okay for now, based on what we can see. Maybe we could salvage that. These are things, though, that are going to be hard and are going to take a lot of proof. What we use first is do it in mice, and then if it works, then we would say, well, what could we do it in a human? So new therapies will need to take into account what are the genes that are involved. And it'll take time for new therapies to get developed. But even if we get to that point, it'll still maybe only useful for a portion of kids that have this, because we'll have to know what they have that's wrong, and then we have to correct it. So this is not easy. So when parents come to me and they say, will we have a treatment for this in 5 years? I never say yes, because 5 years is just a very short time for us to do a whole lot of stuff. So we're talking a long time, and that's just the reality of things. And so I wish it was faster, but it just takes us time. Can we help? So this is a really good question, and I have a lot of parents that ask me this: how can I help? Like, how can I make things different? Even if you can't say that you can find a cure or a therapy, can I help somebody? One of the ways to help is that. Oops. Uh-oh, I'm stuck again. One of the ways you can help is to encourage financial support for 1p36 research. I was talking with another family, and we were talking about how there are groups that seem to get a lot of funding, and there's other ones that don't get so much. All of us have to dedicate our lives as researchers to something. And so one of the things that narrows or points people into an area is that you can actually get funding to do that research. It kind of doesn't make sense that, well, shouldn't you just do research on everything? And the answer is we just can't pay for that, of course. So if it means something to a whole bunch of people, well, that's when government tries to do things and help. But there's also a lot of things that can sometimes just be done by families. And so one of the things that we always are looking for is, are there individuals that want to support this kind of research financially, or are there groups that would be interested in raising money to support research for this? I work on lots of different diseases. One of the diseases I work on is diaphragmatic hernia, which is basically kids that have holes in their diaphragm, or that muscle between their abdomen and their thorax. I go to talk to their parents' meetings too. And they say, hey, you know what? This is a bigger problem than most people realize. And the answer is, yeah. Probably none of you have ever heard of diaphragmatic hernia, but it's twice as common as 1p36 deletions. And these kids die on a regular basis. And so those parents think that they're the most important thing in the whole world. I mean, we ought to conquer this. And I'd say, yeah, I mean, yes, I work on that too, right? So sometimes parents have actually come together and they raise money and then they give that to research. That's another way to do things. But even if you can just encourage research for scientific research in general, that could be helpful. Because right now there's less and less of that money around. So that'd be one way to help. The other thing is to become personally involved in research projects. So for example, I talked about a lot of things where if we had all the information, we'd be able to figure things out more, right? And so one of the easiest things to do is be part of a mapping project. So I work on mapping projects, and if you'd be interested in doing that, we would love to have you. The cool thing about a mapping project is it doesn't take much more information than what you already have. So in this particular case, what do we ask for our families? We ask for their microarray test result and then a clinical summary. And that's sufficient for us. So we don't do blood samples or anything else. Instead, we take the data that's already there and we figure things out. But to do that, I mean, all it takes is people who want to be part of this. One of the things you can do is you can email me if you want to learn more. I also put out there's like 2 little clipboards. They're on the outside. If you want to put down your name and your phone number and your email address, we'll email you and we'll tell you all about what we do and see if you want to be a part. Most parents are actually really shocked that it doesn't take a blood sample and like 7 years and all this. It's really simple. Most of this we do actually over email. We'll just contact you, we tell you about what we do, explain it, and then you can decide if you want to be part. And hopefully there'll be many more things like this that will be coming in the future. hopefully including things like clinical trials and such. We would just encourage you to be part. Stuck again. So that's basically the end of my talk. There's some important people I should mention. So all the people in my lab that work on this project are shown up here. So there's a bunch of us. But again, we're just a small group. When you think about it, the number of people who work on this is very small. But this is us. There's other people who helped us with the mouse models that we use. This is a person who helped figure out what the gene was. This is the person who gave me the mice. The other thing we get is we get funding from different sources. So the Carolyn Weiss Law Fund, basically this is a private person who says they want to do medical research on genetics, and so they gave us money to do this. And then this is money from NIH, which is the National Institutes of Health, so that's basically our government. So when people say, well, who do you work for? I work for Baylor, but the answer is I also work for you. Because every time you pay tax dollars, part of that comes to me and my lab. So we want to do the best we can for you. And we are absolutely dedicated to doing this. This will be something we will work on for as long as I'm alive. Thank you much.